A good day to all. Today's topic of discussion is the differences between mucous SNA and serous SNA. Now, serous SNA are secretory units which are seen in serous salivary glands. The mucous SNA are the secretory units which are seen in the mucous salivary glands. Say, for example, parotid gland is considered, or it is the serous type of salivary gland. Submandibular gland is a mixed type of seromucous or mixed type of salivary gland, whereas sublingual gland is a predominantly mucous type of salivary. And all the minor salivary glands, most of the minor salivary glands are considered as the glands with mucous type of secretions. Now we are going to see what are the structural differences you see when you look at the serous and the mucous SNA. And what are the differences in its function? And what are the differences in the contents which these SNAs have? Now, when you take the shape, the serous SNA are spherical in shape. As you see in the picture, it is more round or spherical in size. And it is comparatively smaller in size when you compare to mucus. And the serous SNA will have a smaller lumen, or the central lumen of the serous SNA is going to be smaller when you compare it to the mucous SNA. Now, what are these lumens? Are the central small empty areas onto which the secretions are being pushed into. And these lumens will continue as ductal system of salivary glands. The lumen will continue and form the intercalated duct, and then it will continue as a striated duct, and then as an excretory duct, and through which these secretions, which is basically coming out of the serous or the mucous cells, being pushed out into the oral cavity. And it also, the serous SNA will also contain intercellular. These small pyramidal shapes are called as serous cells. Say, for example, in this first picture, serous is the whole unit is called serous SNA, where it has 1, 2, 3, 4, 5, and 6 or 5 with 5 nucleus. So 5 individual serous cells is making up this SNA. And the outermost small cells, what you're seeing, the nucleus is seeing, is the myoepithelial cells. Okay, so now in between these cells also you can see some canaliculae in serous SNA alone. Okay, you don't see it in mucous SNA. And the secretions will have these, the secretions what is coming out of the serous SNA will have lot of enzymatic activity, which will contain, by virtue of the presence of acid phosphatase, esterase, and all those contents are there. So it will have a lot of enzyme activity, which is mainly necessary for the primary digestion. Some of this amylase and some components are immediately will start getting digested in the oral cavity itself. Only then it is being taken into the gastrointestinal tract for the further digestion. So some of the smaller molecules are even gets digested within the oral cavity itself because of this presence of these enzymes in the serous secretions. And these serous SNA will secrete secretory proteins, and carbohydrate contents are minimal because most of the carbohydrates contents are usually seen in mucous type of secretions. Okay, so and then now these are some of the shape, and when you look into each cell, the serous cells are pyramidal in shape. As you can see in this picture, the apex of the cell is more narrower, whereas the base is more broader. So that hence it is making a pyramidal shape of serous cell. And it is, as you see in the diagram, it is having a spherical or a round nucleus. And where is the nucleus placed? It is more placed in the basal. Now, base means that broader area, which is attached to the outermost membrane, is considered as a broader area. And the apex is towards the lumen. Near the lumen, what you see is considered as the apex of the serous cell. The whole thing is called as an SNA, and individual pyramidal units are called as cells. And you can see the nucleus is made up of spherical or round nucleus, which is present in the basal layer. Why it is seen in the basal layer? Because all these contents has to be expelled out into the lumen. So you have a lot of contents getting the vacuoles or small vesicles which contains proteins are getting accumulated on the basal or in the apical region. Sorry, not on the basal region, in the apical region. So hence this nucleus is being pushed towards the basal area. And the apical portion will contain lot of zymogen granules, and hence this, when you stain the serous cells, the serous cells will take maximum color or stain when you compare to the mucus. The mucus is unlike serous, it is more pale in color. Why it is more stained? Because it contains a lot of zymogen granules which is present on the apical portion. Hence it is stained strongly by hematoxylin and eosin stains. And the apical portion shows numerous eosinophilic secretory granules, which can also be visible in HND stain, and also by some special stains like toluidine blue stains. So these particles or these granules can be properly visualized by using some special stains also. Now we will move on to the mucus acini. The mucus acini is more elliptical in shape, or it is more oval. It is not exactly round, it's slightly towards oval, or it is elliptical in shape. It is larger in size when you compare to the serous acini, which is relatively smaller. And it contains a larger or big lumen when you compare it to serous. I told you the serous will have smaller lumen because this is more pyramidal shaped cells are there as serous cells. So the lumen is going to be much smaller, whereas in mucus, the lumen is more larger. And in between these cells, this mucus cells, you don't have intercellular canaliculae, whereas in serous, you saw some intercellular canaliculae as well. The secretion, the type of secretion which comes out of mucus is more thick, it is more viscous, it contains more of carbohydrates, and the enzymatic activity is almost nil. It doesn't contain any enzymes which are capable of digesting any protein or any carbohydrates. So the secretions will not have any enzymatic activities. This will only produce more carbohydrates condensed than proteins. And when you look into each cell, this mucus cells are also pyramidal, okay, but not as pyramidal as like your serous, but this is also pyramidal. The nucleus is more flattened. It is not exactly a round or large nucleus like serous. The nucleus is more flattened and it is placed towards the base of the cell. The apical portion of the cell appears empty, whereas in serous you were seeing a lot of granules and zymogen granules which is present in the apical portion, but whereas in the mucus cell the apical portion is relatively empty. So the apical portion, the staining, the whatever staining which is taken by the mucus cells are relatively pale because there is not much contents in the apical portion or in any part of the cell as such. And the apical portion can be stained strongly with carbohydrate stains because the cells will contain full of carbohydrates, mucopolysaccharides or carbohydrates. So it can be visible properly if you do special stain like periodic acid shift stain or PA stain, or you can put Asian blue stain. So these are some of the stains which can be used specifically for carbohydrates. So this, if you want to see specifically what content is there in these empty areas, you can put some special stains like Asian blue and PAs, and you'll be able to visualize this carbohydrates which is present, because the HND or the hematoxylin and eosin is going to be very, very pale in mucus as such. So that is the main differences between mucus and serous acini. And if you go into a little bit of detail, some ultra microscopic structures also you can discuss or you can write. But this is basically, which is enough for the differences between a mucus acini and the serous acini. When you prepare an answer for this mucus or serous acini, you can draw a neat label diagram, which is mandatory, and you have to label it specifically. And you can make two tabular columns, and you can describe it as what mucus cell, like what is the shape, what is the size, how the nucleus is present, what are the contents of mucus cell and serous cell separately. And you can write separately in separate column what are the shape and the size of a mucus acini acid. Now the acini, the complete unit, what you see in this diagram is called as acini, and what you see separately within it is considered as individual cells. So you can explain acini, the structure of acini separately, and the structure and the shape and the content of cells separately when a question is asked what is the differences between serous and mucus acini. With that, we will end this short topic. Thank you.